This is a journey into the molecular world of a vitamin that often flies under the radar, but it is absolutely powerful. This is vitamin K. We'll start with imagining a child who falls during a gully cricket match. He scrapes his knee. What's the immediate life-saving response? Well, a clot forms, right? But vitamin K is not just about clotting. Whenever we think about clots, most of the time in medicine we think it's related to vitamin K. Vitamin K is much more. It conducts a symphony of processes from bone strength to arterial health. Yeah, it's pretty capable. It's a classic example of how a single cofactor can unlock an entire cascade of vital biological function. So let's dive into it, shall we? It's a high yield topic for many exams, and more importantly for future patients in India. I would like to start with the key concepts. So the key concepts is the forms and why they matter. There are different forms of vitamin K. So the first thing we talk about is the forms. Vitamin K is not just a single entity. It's a group of fat-soluble naphthoquinone derivatives. And when we talk about vitamin K, we talk about mainly three forms. The first one is vitamin K1. The second one is vitamin K2. And there is a K3. The first one, which is vitamin K1, is also called phylloquinone. So phylloquinone is found primarily in green leafy vegetables like spinach, kale, broccoli, and even some vegetable oils. When we talk about the clinical relevance, this is your primary dietary source crucial for liver-centric coagulation factor synthesis. So it's essential for coagulation factor synthesis in the liver. Think of K1 as the liver's favorite child for clotting. Then we have vitamin K2. Now these are called Menaquinones. This is a diverse group. Some forms are synthesized by our own friendly gut bacteria, your internal production unit. Others are found in fermented foods like natto. Have you heard about natto? It is less common in India. But think fermented idli dosa batter. Though not directly a source, but it is a source. Other than that, there is cheese and some animal products. And some forms have also been known to be formed endogenously. The clinical relevance is it's increasingly recognized for its role in extrahepatic tissues. Anything other than the liver is extrahepatic, especially bone and cardiovascular health. K2 is the silent guardian of your arteries and bones. Then we have vitamin K3. This is a synthetic form, and it is the only form which is water soluble. In the beginning, we saw how most of them are fat soluble, but it's the only form which is water soluble. And the clinical relevance lies in that historically it was used, but due to potential toxicity, they have found that due to menadione, there was hemolytic anemia and liver damage. So it is no longer used therapeutically. We mention it for like completeness, but your focus should be vitamin K1 and K2. Now, sure, we have talked about these different forms, but a student asks me, why is this relevant, sir? Why do we need to know the distinction? Okay, at the end, it's vitamin K, and in clinical practice, we just go with it, right? We just go with the supplementation. Now, why it matters is because K1 and K2, both of these, have different dietary sources. They have different dietary sources as well as different absorption pathways and different tissue distribution, which influences their specific roles in the body. It's like different types of trains on different routes, all part of the same railway network, but serving distinct purposes. So it's a valuable concept which we need to understand in biochemistry. Next, we come to the absorption and transport. I like to call this the journey from plate to plasma. So imagine you have eaten your palak paneer, which is rich in K1, or some cheese, which is rich in K2. How does this fat-soluble hero get into your system? It's a classic lipid absorption entry. So we recall our fat digestion. First one is diet intake. So the dietary intake occurs K1 from veggies and K2 from gut bacteria or animal products. The second one is digestion and micelle formation. This happens in the stomach and small intestine. So it occurs in the stomach and small intestine. And the bile salts, which are, if you recall, they are produced in the liver. From the liver, they are stored in the gallbladder, which is crucial for fat absorption. These emulsify dietary fats. Pancreatic lipases then break them down. Vitamin K, being hydrophobic, is like a VIP passenger needing a special ride. It gets incorporated into these tiny spherical transport vehicles called mixed micelles. So there is emulsification of fats and then formation of mixed micelles. So if you recall, the micelles are structures like this. Our vitamin K, being hydrophobic, is like a VIP passenger needing a special ride. So it gets incorporated into these tiny spherical transport vehicles called mixed micelles. Think of bile salts as the taxi service for fat-soluble vitamins. If your patient has a condition affecting bile production or flow, example obstructive jaundice, cholestasis, or even pancreatic insufficiency, then vitamin K absorption will be severely impaired. This is a common exam scenario, so just keep that in mind. The third step is enterocyte absorption. So now these micelles ferry vitamin K to the brush border of your enterocytes. These go till the brush border of your enterocytes. So these micelles reach the enterocytes near the brush border, and vitamin K now can passively diffuse across the enterocyte membrane. Once the enterocyte absorption has happened, the fourth step begins. The fourth step is chylomicron formation. Inside the enterocytes, our vitamin K is now packaged along with other dietary lipids into larger lipoprotein particles called chylomicrons. These are like the long distance buses of the lipid world. The fifth step is lymphatic transport. Chylomicrons are too large to directly enter the bloodstream, so they take the scenic route via the lymphatic system. Eventually, they drain into the bloodstream through something called the thoracic duct. So at this point, our vitamin K from the diet has come into the bloodstream. That is a big achievement for us. What happens now is the sixth step, which is liver uptake and distribution. Chylomicrons circulate, delivering fats to peripheral tissues, then taken up by the liver. If you recall the role of chylomicrons, they mostly take fatty acids to the peripheral tissues. So they just do all of their job, and their remnants, once they have become smaller in size after giving the fatty acids to different peripheral tissues, they come to the liver and they are taken up by the liver. Now these chylomicrons still carry vitamin K. Vitamin K now enters the liver. Liver is the primary processing and storage hub. Though its storage capacity for vitamin K is a bit low compared to, say, vitamin A. From the liver, vitamin K is then repackaged into VLDL and subsequently transferred to LDL for distribution to extrahepatic tissues. So inside here there is transport to VLDL and then LDL, and LDL takes it to extrahepatic sites, bone and arterial wall, like we discussed earlier. This efficient system, so if you just look at this entire system, this is a very efficient system, and it ensures our crucial cofactor reaches its various destinations across the body. Now we come to concept of the vitamin K cycle. This is where the real biochemical magic occurs, in the vitamin K cycle. The vitamin K earns its stripes as a cofactor here, and it enables a unique post-translation modification called Gamma-carboxylation. This isn't just a fancy name, it's the key to activating many vital proteins. This is a very important step. Let's talk about the vitamin K cycle for a bit. First one is the substrates. We have an inactive precursor protein. So this is how it works. We have an inactive precursor protein which is loaded with glutamate residues. These are the raw materials waiting to be processed. We also need carbon dioxide and the active form of vitamin K, which is vitamin KH2. So we need a precursor protein which has lots of glutamate residues. These are the raw materials waiting to be processed. We also need carbon dioxide and the active form of vitamin K, it is also called KH2, or vitamin K hydroquinone, or it can also be called as vitamin K quinol. The next step is carboxylation reaction. First we discuss the substrates, then the carboxylation reaction. This is the main event. The pivotal step is catalyzed by this special enzyme which is gamma-glutamyl carboxylase, or in short GGCX. Remember this enzyme, it is very important. So the mechanism is that KH2 isn't just a passive observer, it acts as a reductant, donating electrons to activate CO2 and facilitate its addition to the gamma carbon of the glutamate residues. In this process, KH2 is oxidized to KO, vitamin K epoxide, which is the inactive form. The glutamate residue is transformed into gamma carboxylate. So this becomes gamma-carboxylate. each glutamic acid residue now has 2 carboxyl groups, making it super efficient as a chelator for calcium ions. Think of GLA or glutamic acid residues as the Velcro for calcium. The third step is regeneration of vitamin K. This is the recycling step. The oxidized vitamin K epoxide must be recycled back to active form, right? For the cycle to continue. So this is a two-step reduction process. Step A is epoxide to quinone. This is the step A. Vitamin K epoxide reductase is the main player here. It reduces KO back to vitamin K. This enzyme, which is vitamin K epoxide reductase, is the primary target of the anticoagulant drug, which is warfarin. Warfarin basically blocks the action of this enzyme. Then there is step B, which is quinone forms hydroquinone. Vitamin K quinone is reduced to the active vitamin K hydroquinone, which is KH2, by either NADPH dependent quinone reductase or a dithiol dependent reductase. This elegant cycle ensures that a relatively small amount of vitamin K can continuously facilitate the carboxylation of numerous proteins. Even small amount is quite enough. There's no direct ATP yield here. Instead, vitamin K itself is the energy currency for this specific carboxylation reaction, with its redox state driving the chemistry. It's like a closed loop recycling plant